Hello, dears, and welcome to Al-Hussaini Virtual Lab, Pathology Talks, Tips, and Practical Tips. Today, I'm going to share with you an interesting case of atypical meningioma and how we can use Ki-67 in particular to locate mitosis and start our counting. So this is a case of a 12-year-old female patient who has been known for medulloblastoma, treated for medulloblastoma several years ago. Now she developed an extraaxial lesion, and the excisional lesion, as you can see, has some features reminiscent of a meningioma. There is certainly a tendency at whirling pattern, and the tumor is punctated by many lymphocytes, and there are areas of hemorrhage. Now, at high power magnification, perhaps this image will be more aligned with meningioma, where we have syncytial growth pattern. And what do we mean by syncytial growth pattern? That the boundaries between the cells are indiscrete. So we cannot really draw a line around the end of the cytoplasm of one cell and the beginning of the cytoplasm of the next cell. This is what is meant by syncytial growth pattern. Perhaps some tendency or something similar to whirling pattern, but it's not really very well clear. Neither are really intranuclear inclusions, but this syncytial growth pattern should be really very helpful. Now remember that atypia in meningioma is not important. So pleomorphism is not one of the features that we look for while we sign out cases of meningioma when we want to assign the grade. We look for other features like the sheety growth pattern, the prominent nucleoli, necrosis, a small cell type. If 3 out of these features are present, we can assign a case as atypical meningioma. However, pleomorphism is not one of those features. Now, I'll be talking about the other features of atypical meningioma while we proceed with the presentation. So we performed EMA stain, and remember that EMA stain is positive in meningioma. It tends to show weak to moderate staining usually, and it has this wisp kind of positivity highlighting the cytoplasm and the cytoplasmic processes. This is in contrast, for example, to ependymoma, where we have the dot-like cytoplasmic staining with EMA. So we have to really to pay attention to the pattern of immunostain. It's not only positive or negative staining. And then we have the PR staining, which is positive, shows nuclear staining in some of the tumor cell nuclei. Both of these would support that this is a case of meningioma. And to sort out the other feature that we depend on in cases of atypical meningioma, which is brain invasion, I almost always performed a GFAP immunohistochemistry to look for and then decide whether there is a brain invasion or not. And in one previous tip, I showed you a typical example of a brain invasion in meningioma and how the GFAP would be very helpful in sorting out this important feature that would automatically upgrade the tumor, the meningioma, into grade 2 or atypical meningioma. So in this case, the outlines really are kind of smooth. We don't see islands of meningeal cells within the GFAP positive brain parenchyma. So this would support that there is no evidence of a brain invasion. What I always do actually with meningioma is Ki-67. And Ki-67 tend to show this variability of immune staining. There are hotspots, there are less positive areas. So it's really important to locate your hotspots for 2 reasons. Number one is that there is some evidence that if Ki-67 is more than 4% in meningiomas, in hotspot, these meningiomas tend to behave like atypical meningiomas. So this is number one. Number two, what I really do in practice is when I locate my spots, I go on high power magnification and I start really looking into the presence of mitosis. So this is a nucleus that is Ki67 positive, another one, a third, a fourth, and so on. But this one here is actually mitosis, because as you can see with Ki67, whenever we have mitosis, there is a tendency to stain the cytoplasm as well as the chromatin within the nucleus. And there is another mitotic figure in the hotspot. So I really sometimes, if I'm desperate, I go and count mitosis. I kind of cheat. I count mitosis in Ki67. But what I usually do is when I locate my hotspots, I go back and mark this area on the H&E slide and look for mitosis in that particular area. And in this particular area, I started to locate mitosis. This was one mitotic activity. And just in the field following that, there were two mitotic figures and a third and a fourth. So I ended up in that hotspot counting on the H&E 6 mitotic figures per 10 high power fields, which would be consistent with atypical meningioma. So the final diagnosis for this case was atypical meningioma, CNS WHO grade 2, based on the increased mitotic activity in excess of 4 mitoses per 10 high power fields, as well as more than 4% Ki67, which some literature would really support that this kind of meningiomas behave in a more aggressive fashion. So remember always to use Ki67 in your practice in meningiomas. To number one, try to estimate the Ki67 proliferative index, and number two, try to find your hotspots, and on those hotspots you can either count the mitosis directly on the Ki67 or mark the area, the same area on the H&E, and start your mitotic count. And believe me, it works on very well. I hope you find this tip useful in your daily practice. Thank you.